Yeah. So folks are still trickling in, and Ben is still getting his microphone up and working. But welcome everyone. This is our end of the year webinar for our Sierra to Sea program, which ran in 2019. For those of you who aren't familiar with this program, it's been running since 2015. And in that span of time, we've mobilized 25 citizen science events, engaging over 210 participants with 7 partner organizations and surveying over 23 meadows, which is really incredible. This year alone, we had over 25 participants out on citizen science events, 4 different events, which were all really accessible, surveying 4 different meadows and collecting data that we otherwise wouldn't have been able to collect. If you're not as familiar with this program, the purpose of this program is to build climate change resilience by decreasing greenhouse gas emissions, increasing water availability, and conserving biodiversity in the meadows of the Sierra Nevada mountains. And by using a citizen science program specifically, we're able to mitigate and adapt to climate change in California, and by incorporating citizen science, increase the volume of data collected and fill in gaps critical to our understanding of how these meadows affect the regional carbon cycle. Joining us to talk today are Ben Sullivan, Aaron Smith, and Rachel Hutchison. Ben and Aaron are both coming from the University of Nevada, Reno, where Ben is a professor, and Aaron is works with Ben in his lab. And Rachel is coming from the South Yuba River Citizens League, or SYRCL. So welcome both. In terms of our schedule today, we're gonna have Ben and Aaron present on their data from 2019 and their findings for about 15 to 20 minutes. Then Rachel will be talking about the importance of citizen science and the work SYRCL does for about 10 to 15 minutes. And then we'll use whatever time we have left, hopefully about 15 minutes for question and answer. If you want to submit a question, you should see a Q&A button at the bottom of your Zoom screen, and you can submit questions there. If you're having trouble with that, feel free to send them in the Zoom webinar chat, and I can pass them along that way. So if everyone's ready, and if Ben, if your microphone's working now, we can get started with your presentation. I'm going to share my screen, and I'll be clicking slides for Ben and Aaron.
Actually, Ian, I think I've got this going. Can you hear me?
Yeah, I can hear you.
Great. So I might be able to share my screen here. I think I can share this and bring up our slideshow.
Wonderful. Then I will. I'll mute myself and I'll leave you to it.
Oh dear. Let's see. This appears to be reversed. Swap displays. There we go. Okay. So just to confirm, you guys should just see a title slide. Is that correct? No additional slides?
That's what we're seeing.
Super. Okay. Well, it's great to have an opportunity to share the results of all our hard work from this past year. Welcome back, everyone. We haven't been able to see you yet, but it's good to see you. So I wanted to start this off by just talking very briefly about what we were able to accomplish as groups this past summer. We managed to visit 5 meadows, 4 of which we're going to discuss here today because you guys participated in those 4. So those would be Lower Aloni, Upper Aloni, Deer, and Truckee. So those are the ones that we're really going to focus on for the webinar today. By my reckoning, we sampled at least 108 soil cores, which is a pretty phenomenal amount of samples to collect. we collected 324 individual greenhouse gas excitaners. So you guys will remember filling those with gas samples. And so cumulatively, that's what we pulled off. And then in addition, we have 54 samples of vegetation biomass and community composition from different vegetation communities within each meadow. And so each meadow has different veg composition, and so we set this up. We stratified our sampling by the different vegetation communities that were present in each meadow. Okay, so I'm going to give us a quick review of what we were trying to do. We talked about this, with each campaign with you guys. It's been a while now. Aaron and I have been thinking a lot about this lately. So this is all fresh to us, but I figured it would be helpful to go back over it. So in the big picture, what we want to know is whether a meadow is a net carbon source or sink, meaning is it sequestering carbon from the atmosphere as a sink, or is it producing carbon to the atmosphere as a source? Okay, so understanding that depends on knowing how much carbon is sent below ground by plants. So once those two things are sort of broader goals of the work we're doing in my lab. Once we have a sense of how much carbon is allocated below ground, we need to know how much carbon is returned to the atmosphere as CO2 from roots specifically. In order to, it's helpful to remember that yes, plants photosynthesize, they suck CO2 out of the atmosphere, but they respire CO2. They combine the CO2, they combine the sugar that's made in the leaves upstairs above ground in the plant, and then they combine that with oxygen and produce energy in CO2, just the way we're respiring our breakfasts. Hopefully, we had a yummy breakfast, and that's giving us energy to have this webinar. So what we're doing is partitioning the CO2 that comes out of the soil into its two components. The CO2 that's produced by microbes, which we call heterotrophs, because they get their energy from diverse sources, and plants, or roots specifically, and which we call autotrophs because they get their energy by making it themselves through photosynthesis up in their leaves. Okay, so what we were trying to do is with this project, the goal of this project was to break out these two sources of CO2 that come out of the soil into the microbial fraction and the plant root fraction. We described this approach in the field. It's hard to visualize, and so I've got some slides here to try to help explain this, and then we'll be able to actually see it in practice. What we're doing is using a regression approach. It's a statistical model approach to partition out the respiration or the CO2 produced by the heterotrophs, the microbes, and the autotrophs, the roots. Okay. So we first have to go sample the total amount of CO2 that's produced from the soil surface. And you guys did that when you put the chambers on the ground and then clipped out all the vegetation and then measured the CO2 that was coming out just from the soil surface. Okay, so that was easy enough. That took 3 samples, and we measured the CO2 that's produced by the soil. Then we took cores from inside those chambers, right? So inside those soil cores were roots. We create a mathematical model, that's that statistical model, of the relationship between root biomass and soil CO2 flux. And we want to know what the y-intercept is, right? So in y equals mx plus b, we're going to try to solve b. And that is going to help us know how much CO2 is produced if there was no root biomass. right? Let's pretend that you had a soil core that had no roots in it at all. How much CO2 was actually produced? That would be the amount of CO2 that's produced by the microbes, right? Because there are no roots in there to be contributing CO2. And knowing the amount that's produced by microbes then allows us to figure out how much of the total respiration that we saw from the soil surface was from microbes or from roots. Okay, so we can partition those out. So we're doing this because we know this is an important estimate that we need in order to be able to determine if a meadow is a sink or a source. We expected to see a significant fraction of CO2 be produced by roots because we know that there's a lot of root biomass in these meadow sites. So that was sort of our expectation coming into this. However, we know that sites are going to have different fractions of CO2 produced by roots because the sites themselves are fundamentally different, right? So for those of you who got to visit multiple sites, you got to see there's differences in veg communities. Those differences in veg communities are associated with fundamental differences in depth to groundwater and soil water content within the sites, and how those vary. And we also know that roots do other things than just acquire water, right, and produce CO2. So they also absorb nutrients that are necessary for plants, and so there might be differences in nutrient limitation and things like that that could influence why there are roots in the core and what the roots are doing, okay? So one of the reasons I was really highly motivated to do this work is that we tried this approach before using sort of a coarse estimate. And basically what we did was we had soil cores that people had collected in meadows, and we had CO2, soil CO2 flux rates, but they were never taken in the same location or on the same day and things like that. And so when we had used this regression approach, we did not actually find a significant fraction of CO2 was produced by roots. And that kind of made me think, okay, there's a reason why that's possible, but I'm not sure I believe it. And I want to do this better, right? So we designed this project that you guys were able to help us pull off. Okay, so what does all this regression stuff look like? Well, it should look like a scatter plot. And this is when I say should, this is sort of my idealized interpretation of what this would look like. We have root biomass on our x-axis on the bottom, and we have soil CO2 production on our y-axis on the left. Okay, and if we had some ideal relationship, what we might see is that as we increase root biomass, we have an increase in the total amount of soil CO2 that's produced, and we can fit a line through that. Okay, so we've got data points that are represented here, and then we can fit a line through those data points. We can then estimate what the y-intercept of that line is. And here I've just sort of drawn roughly where it might be with that red arrow. And if that is actually our y-intercept, we can say, okay, that's the y-intercept. And then the difference between the y-intercept and the data point, as shown by these two brackets, represent the amount of root-derived CO2 that's produced, and anything below the y-intercept represents microbe-derived CO2. Okay, so in an idealized world, this is what our data would look like, and unfortunately, nature is not always so predictable. So this is what we're hoping to see, and what we're about to show you now is what we actually found. Okay, so this is kind of how it should work theoretically, and so we can then say, okay, when there's not very much root biomass, there's sort of a uniform amount of microbially derived CO2 that's being produced. We have a relatively small amount of root derived CO2. But on the right-hand side of that x-axis, when you have a lot of root biomass, you're going to have the same amount of microbially derived CO2 that's produced, but you have a lot more root-derived CO2. So we can use this to basically say, all right, like what fraction of soil CO2 that's produced is actually coming from roots versus microbes. Okay. So after we split, right, and you guys went home and we went back to Reno, there was a lot more work to be done, processing all those soil cores and sieving out the roots and washing them off and weighing them and drying them was quite a bit of work. And then there was more work on the statistical analysis side. What we found was kind of what we expected, which is that each site was a little bit different. So I'm going to show you guys each site. And some of you visited several of these sites, and some of you probably just visited one of these sites. So we can talk about those later if you have questions as we go through in detail here. Okay. So first up on our list is Lower Aloni, which I think was actually the first one that you visited. So here's the relationship between root mass on the x-axis and the soil CO2 flux that you guys measured in those static chambers after clipping the vegetation on the y-axis. And what we see here is that we have data points. These are the dots. The dots are colored by vegetation type. So Aloni, we identified 3 different vegetation types in Aloni: wet, mesic, and mesic. And then we fit a statistical model to all the data, not just one vegetation type. And what we see is that we have a pretty much a flat line, right? There's no strong relationship between root mass and soil CO2 flux. Okay, so that's telling us that for per unit increase in soil root mass, we don't really see a whole lot of variation in soil CO2 flux that would be associated with that change. Okay. But we were able to estimate this y-intercept. So you'll see about a half a black dot right below the 10 on the y-intercept. And that represents, or on the y-axis, excuse me, the 10 on the y-axis. So that black dot, or the half black dot, represents the y-intercept. And the gray box represents the range of uncertainty that we have around that y-intercept. Okay, so our statistical model is telling us that the y-intercept is roughly 10, a little below 10, but it could be higher and it could be lower. So that's what we're looking at in that gray box. So that's the possible range of microbially produced CO2. Okay, and there's no statistical relationship between root biomass and CO2 that's produced. Okay. When we switch to Upper Aloni, which, as you guys will recall, was very wet, there was only wet and wet mesic vegetation from that site that we sampled. And again, we have the same relationship here with root mass and soil CO2 flux. And we have a y-intercept that is a little over 5 on the y-axis. There's a half a black dot there. And again, we have the gray bar that represents our uncertainty bounds around that y-intercept. Okay. So here, there was still no statistical relationship between roots and CO2. Okay. So even though we have that line fit there, it's really just telling us that it's a flat line, that there's no relationship between root mass and CO2 flux. But that's okay. The y-intercept was significantly different from 0, so that's telling us that there's a contribution here that is microbial, and it's a lot. Okay. So then we can switch to Deer Meadow. So for those of you who hiked into Deer with us, that was a long hike and we had to carry a lot of gear. And so thank you for your patience with that. And again, what we have here is that there's no statistical relationship between root biomass and CO2 flux, just as in the last two sites. That's okay, because we have this y-intercept value that we're looking for that's just above 3 with a range of uncertainty. And this range of uncertainty is actually a good bit smaller than it was at the last two sites. So we're starting to feel a little bit more confident in our estimates of the y-intercept at Deer Meadow. Okay. Next was Truckee. And so Truckee had a wide range of vegetation sites, of vegetation communities. So we had some really dry vegetation and some nice wet vegetation. And we actually have a significant relationship between roots and CO2 production. So as you increase soil root mass, you get more soil CO2 flux. So that's the first time we've seen this. Out of the 4 sites we're seeing today, this is the only one that has that significant relationship. And again, we see a significant y-intercept. That's that black dot on the half black dot on the y-axis. It's just above the 2.5, between 2.5 and 3. And the gray bar represents the range of uncertainty again. And this range of uncertainty is the smallest uncertainty of any of the sites that we measured. So this is the one that we're most confident in when we say this number is our y-intercept. Okay, so that's our range of microbially produced CO2. So our confidence in our estimates of microbial versus root CO2 varies by site. We have low confidence in our fraction of microbially or root produced CO2 at Lower Aloni and Upper Aloni. We have higher confidence in Deer and Truckee. So we can rearrange this mathematically and look at this a little bit in a different way. And we can take these y-intercepts and we can say, okay, let's use these y-intercepts, and then let's go figure out what fraction of the actual respiration we saw was due to roots. Okay, so we have the 4 sites: Deer, Lower Aloni, Upper Aloni, and Truckee now along the x-axis. They're broken up by their vegetation groups. Remember, not all the sites had all the vegetation groups. And then we have percent autotrophic respiration on the y-axis. That's the percent root respiration. Okay. Almost always, roots produce less than 50% of the CO2 in these sites. Okay, so whatever the veg community is and whatever site we're in, roots usually produce less than 50% of the CO2. That's really important to know. Okay, and sometimes roots aren't really doing anything, and microbes seem to be doing all the work, producing CO2. Okay. However, we're scientists and we're sort of skeptical by nature, and so we don't necessarily trust our y-intercepts because we recognize that there's uncertainty around those y-intercepts. So we can do the same exact thing as we just did, but with a low estimate of the y-intercept, and that's on the left panel. And what we can see is that now we have the same dashed line representing 50% autotrophic respiration, and we can see that basically everything falls below that 50% line. And then we can use our high estimate of our y-intercepts, and we can see that with our high estimate, roots roots are more important, not more or less important. Roots are more important, but still usually less than 50%. Okay, so on the whole, while there are a lot of roots in these meadows, a lot of the CO2 that's being produced is actually from microbes, and that's a really important finding. Somewhere between, it can vary basically between 0 and 50%, but a more reasonable estimate would probably be somewhere between 15% and 45% of, depending on where we are and what veg community and stuff like that, of total respiration is actually coming from roots. So a major fraction of the CO2 that's coming out of the soil surface is coming from microbes. That's a really important thing for us to know, because what it tells us is that these meadows are producing a lot of CO2, and a lot of that CO2 is coming from the soil organic matter. And that soil organic matter may be old soil organic matter that represents a loss of soil carbon to the atmosphere. Okay, so when it comes to quantifying whether roots are net sinks or sources, this alone does not tell us, but it does become really important in helping us make that estimate, because it allows us to say, oh, microbes are producing a lot of CO2 here, and that microbially derived CO2 may be coming from soil organic matter that was sequestered in the past under different conditions. Maybe when the meadow is wetter, maybe when there was more, wetter climate or something like that. So I'm going to, that's basically all the results we have. I'd like to give Erin an opportunity just to speak about how, her experiences working with you guys, and kind of what this means for her thesis.
Hey, Ben.
Yeah, I'd like to thank
Erin.
Yeah.
I just wanted to make sure Rachel had time to talk, and then actually, what?
Oh, okay.
And then we can come back to you, Erin, if that's all right.
Absolutely.
Okay, wonderful. So, Rachel, we're having a little trouble hearing you. What we could also do is, Erin, you could talk. You could go 5 minutes and talk, and while Rachel just gets her microphone up and running. Does that sound all right?
Perfect. Well, first of all, I'd like to thank all the volunteers that helped out during the course of the whole summer and fall, and Earthwatch. It was definitely very fun. I mean, it's already something I very much enjoy, and that just added extra fun to it. Really, the volunteers were very helpful. We had a lot of different volunteers from a lot of different walks of life, and we also had student volunteers, and all those volunteers really helped us to collect more data that's so important in our findings. I definitely could not have done that myself, or even with other lab partners. We really needed everyone involved, and it was just so helpful. People came out and they had great attitudes. They were so willing to learn something really new, really different, and difficult. It wasn't easy work, and definitely required a lot of heavy lifting and a lot of patience and a lot of attention to detail, and they were really excellent at it. So citizen scientists were essential and the main part of my graduate work here. I also really strongly believe in it, because I think that citizen science and service learning are hand in hand, and it's an important way for our community to really understand the world that they live in and to give back. And when they go home, they can go and talk to people that they know about it and pass it on. It never dies. It's really important. So, and it was just fun. So it was
That helps.
Yeah, it helped. It definitely helped. So,
Great.
Yeah.
Thank you, Erin.
Yeah.
Awesome. Thanks, Erin. All right, Rachel.
Are you able to hear me?
Yeah.
Okay, great.
Yeah.
Awesome. Okay, so I'm also going to try to figure out how to share my screen.
So you should see the green button that says share at the bottom of the screen.
Okay. Awesome. Let's see. Okay, can you guys see that?
Not yet.
Not yet?
There we go.
All right. Okay, I'm gonna do a quick version so that we have enough time for questions at the end. I just wanted to give an overview of some of the meadow restoration work SYRCL's been doing, and the monitoring work that we've been partnering with Earthwatch on for the last 5 or 6 years. So we're a watershed-based organization. This is our territory. It's the Yuba River watershed. Many of you are probably already familiar with SYRCL. Our mission is to unite the community to protect and restore the Yuba River. As part of that, it looks like I'm on autoplay. That's fine. It'll keep us moving. As part of that mission, we have been partnering with Earthwatch, and we also bring a lot of local volunteers out to work on our projects, either to help us monitor or to help us actually do the restoration of all sorts of different habitats. In our headwaters, we work on meadow restoration, we work on forest restoration projects, and then we also remove invasive species across the Tahoe National Forest. And the final thing that we do all the way down at the bottom of the watershed is restore salmon habitat and floodplain habitat. Like Aaron just said, working with volunteers is extremely fun and has been some of the most rewarding work that we've been able to take on here for a lot of the same reasons Aaron said, so I will not repeat them. Here's a look at the Yuba River watershed from within California, just to give you an idea of where exactly it is and where it sits. I would have gone on a little bit more about this one, but I'm going to skip over it. In the Yuba watershed, we have almost 300 meadows that have been mapped by the Tahoe National Forest and others. And that's something that we see as a priority habitat for restoration. And we've also identified several meadows that should be restored in the near term. This is a map of all of the meadows, and some of which many of you have been out onto at Aloni, Upper Aloni, and Deer. Some of you may have also come out to Van Norden Meadow at other times. We also have several projects in the North Yuba and are getting started on some new projects there as well at Chapman Saddle, Haskell Headwaters, Freeman, and Bear Trap. And we're looking forward to continuing to bring volunteers out to those meadows as well. The work that we do in those meadows, I think, you know, the restoration projects, and the reason that Ben and his research team are out doing research is that we're trying to quantify the benefits of restoration and understanding how restoring the stream channel impacts the different types of habitat, the hydrology, the vegetation, and the greenhouse gases in the carbon cycle are all part of what we're trying to learn here. So we have invested a lot of energy over the last couple of years to try and get our meadows to look more like this healthy meadow depiction that American Rivers has put together versus this degraded meadow with eroded stream channels and vegetation that is losing in density. So our main focus has been to look at the restoration response of several different variables. And as part of the work that we've been taking on over the last several years or half a decade, we have been looking at surface and groundwater hydrology, vegetation in both transects and plots, working with Ben's group for greenhouse gas and carbon work. We've been collecting bird samples with point estimates, looking at BMIs and aquatic habitat and aquatic species, and then looking at surface topography. So that's the shortest version that I can do. I don't want to take up too much time, but many of you have been out on our projects, and we hope to continue to encourage you to join us for future monitoring efforts out in the meadows. Zero to see with Earthwatch or otherwise. So thank you.
Thanks, Rachel.
Yep, tried to be really fast.
You were.
Yep.
So before we start questions, I just wanted to say thank you to Ben, Aaron, and Rachel, University of Nevada, Reno, and SYRCL, our funders for this iteration of the program, the Bella Vista Foundation, as well as everyone who was able to come out this summer. It just was a really wonderful experience for me to coordinate this program and get to know this program intimately and everyone involved in it. So thank you. And now we have some questions. and feel free to ask any more questions in our Q&A. So our first question that we have is from Anna, who asked, Ben, what is the next big question? What will you be looking at next as far as research? How does the study of meadows tie back to the larger study of climate change and climate change's impact on nature?
Great question. So let's see. So there's a lot there. How this ties back to our bigger understanding. Well, I think being able to estimate whether a meadow is gaining carbon or losing carbon is really important for several different reasons. One reason is that soil carbon actually represents soil volume, soil mass. And so if you have a lot of carbon in soil, then that represents soil that can hold water. It can filter nutrients and clean water that moves downstream. And so a really important part of the carbon itself is just what it represents physically. Obviously, sequestering carbon in soils has climate change and greenhouse gas impacts, which is originally why the state of California funded the broader question of understanding whether meadows are carbon sinks or sources. And so if we can sequester carbon or meadows can sequester carbon in soil, that represents hopefully a fairly stable sink of carbon that's less vulnerable to fire and insects and things like that than for example, the carbon that's in a forest. So those are some of the big picture things that we're driving at here. In terms of the next steps, I think we're not quite done yet with some of this work that Aaron's undertaken. So we just shared these results last week at the American Geophysical Union meeting in San Francisco with where 35,000 Earth scientists were present. A smaller number actually came by Aaron's poster, but it was a lot, and we got some great feedback. And so I think one of the things that we need to consider is how to build on this specific work. And I'm really curious to know what it would be like if we repeated this work, but did it with some kind of manipulation of the ecosystem. So if we actually watered the soil, within the course of a couple hours, the addition of water to relatively dry meadow soil can have a really profound impact on the amount of CO2 that's coming out. It's not going to profoundly impact the amount of roots because roots aren't able to grow fast enough to respond in a couple hours. And so what I think would be really fun to do is repeat some of this work next year, next summer, but do it with a watering experiment in which we basically start with dry soil, take some measurements of CO2 production, add some water, and then start measuring through time. And then after a period of time, once we think that the water has stimulated root and microbial activity, we can then take cores again. And so I think that'll help us reduce some of this uncertainty that we identified. And so that's I think going to be a fruitful avenue of further work for Aaron.
Awesome. Our next question is from Hita, who asks, do you have any insights into why the meadows studied this summer were releasing more carbon than they were sequestering?
So we aren't able to establish that. We don't know that the meadows were releasing more carbon than they were sequestering. What we do know is that of the carbon that was being released, a lot of it was coming from microbes. So there are other things that we have to do to be able to determine whether the meadow is gaining or losing carbon. And some of that work has been ongoing in these sites. But that's sort of a different angle here. So all we're able to do is say a lot of the CO2 that is produced is coming from microbes. We're not able to say that the meadow is definitively gaining or losing carbon.
Gotcha. Okay. And another question we have is, how can one get involved further locally? And this is for Ben, Aaron, and Rachel as well.
Can I answer first? Is that okay?
Absolutely.
Okay, so locally, I'm guessing in general in the Yuba watershed, and then in the Truckee River watershed, which is where most of these this particular project happened. SYRCL has a variety of volunteer opportunities that generally start in the summer months or in the late spring. That's just because that's when the snow is not present in these meadow systems. And next summer, we are going to be spending a lot of time collecting data in pursuit of understanding the hydrology, the vegetation, and the tree encroachment in several meadows and their impacts to those meadows. And so I would encourage you to sign up for SYRCL's listserv, and we'd be able to get you out in a meadow or somewhere else in the watershed, working on any of those other projects that I mentioned earlier, all throughout the season. We do all kinds of different things. You can come and pull Scotch broom. You can count trees that we planted in the lower watershed in May. We have a lot of different opportunities. And you can actually contact Mary at yubariver.org, and she'll be able to set you up with any of those activities as well. And she's on this webinar, but she's muted.
Awesome. And Ben and Aaron?
Yeah, I can answer that. With the work that we hope to do next field season, you know, we would really encourage and invite people to come back out to some of these meadows that we may return to next season and participate again in this kind of second phase of this ongoing project.
Great. Another question from the students at Forest Charter is, they would like to know more about the relationship of microbes breaking down the dead organic material or roots. So is the carbon being released by microbes still the result of the vegetation?
That's a great question, you guys. All right, so some of the carbon that comes from the microbes is probably material that was recently released from the roots themselves. So this partition between autotroph and heterotroph is a black and white split, which in nature is often trying to make a split in a gray box, right? Trying to partition black from white in a gray box. So an element of the a certain amount of the CO2 that we're calling microbial is probably processed by microbes, but it was recently in a plant. And the plant has sort of released that sugar out of its roots into the soil, perhaps as a way of cultivating a healthy microbial community around its roots, in order to obtain nutrients and do other things that occur in the soil. There can be many mechanisms why a plant would want to control who's living around its roots. But that sugar that's sort of released out of the roots could then be eaten by these microbes and produced as CO2 that we're calling microbial. So one of the things that we do to actually determine whether a meadow is a net sink or source of carbon is we actually feed the plant isotopically labeled carbon. It's a form of heavy carbon dioxide that allows us to be able to trace the carbon dioxide that's used by the plant. And so we can actually get at that. We can figure out how much carbon the plants are sort of leaking into the soil environment and feeding microbes. And by our estimates, it's actually a fairly large fraction of the amount of photosynthesis that occurs. Off the top of my head, I think we're looking at numbers between like 5 and 15% of the total amount of photosynthesis that occurs is actually just like leaked into the soil. And so that's the plant feeding the microbe, and then the microbe producing CO2. So we can actually get at that other ways. But you're spot on. Some of that microbial CO2 is actually recent plant material.
That's a great question. Another one from Rachel Mirowitz. We have, in restoration, how would one go from a bare land to facilitating meadow production, for example, runoff mitigation or native ecosystem support? And Rachel, feel free to chime in as well.
Rachel, do you want to take a whack at that?
Yeah, sure. Sorry, I couldn't remember how to unmute myself. So a lot of the work that we've done to restore meadow ecosystems here has to do with reconnecting the stream to its floodplain. And literally one of the slides I showed, which I didn't go into detail on, what we're trying to do here to increase vegetation production and increase that wet sort of spongy meadow ecosystem is to fill in some channel segments or reroute water in a meadow so that it travels across the floodplains and not just is not just sitting at the bottom of a stream channel that has eroded over the last 150 or so years as a result of what we have concluded are impacts from logging, impacts from grazing, which used to happen at a really high density in some of these meadows before the Forest Service changed their management practices. And what we're doing is filling that in so that as the water melts, as the snow melts and the water starts sort of flowing in these systems, it comes up onto the floodplain and can infiltrate back through into the groundwater, keeping everything really wet. So we're trying to encourage that process in these meadow restoration projects, which in turn, we have this hypothesis that that will increase carbon sequestration over time. And Ben and his team are working really hard, and we worked really hard on trying to figure out how true that is and what the variability is within the answer to that question.
And I can give a
Yeah.
very quick sneak preview of those results. So very briefly, after restoration, the plant community, the above ground plant community responds really quickly, which is something that a lot of people have seen over and over. The vegetation bounces back. Above ground. Below ground, it's a slower story. The roots take longer to sort of recolonize the soil. And so, and as they do, as the roots recolonize the soil, they start producing more carbon. So we actually see a significant increase in soil carbon over the first 20 years after restoration in the top 15 centimeters of soil. But it's a little bit less clear, that pattern is less clear below ground. Below 15 centimeters. So in that very surface soil where the roots are doing the most recolonizing and the most work early on after restoration, that's where we get a lot of our bang for our buck when it comes to carbon post restoration.
Great. Well, I wanted to again thank everyone for attending. And if you'd want to stay involved and stay up to date with the work being done, like Rachel said, you can follow their newsletter or follow SYRCL on Facebook. You can also follow Earthwatch or Sierra to Sea on Facebook to stay up to date on events such as this and future citizen science events that can get you out into the field. And I just wanted to say, again, thanks to Ben, Aaron, and Rachel for coming and telling us about their work and keeping us up to date, as well as our funders, the Bella Vista Foundation, and everyone who was able to get out into the field and help us collect all this data this year.
Yeah, thank you, everyone. I wanted to add one more thing based on a comment that Hitta made in the comment fields. Our Wild and Scenic Film Festival is coming up here in January, and we're lucky enough to actually have there's a film that Earthwatch put together about one of their expeditions out. I think it's in Spain, in the mountains. It's a beautiful film. I watched it recently. That's going to be at our film festival. And then we are organizing a citizen science workshop that will be at 11:30 on Saturday morning. It's a free event. We're hoping to have a 2018 Earthwatch alumni come, or actually not Earthwatch, but one of our high school alumni come, and also Earthwatch will be there. Mark Chandler should be in attendance, per what I understand. So we're really excited about that panel discussion to talk about citizen science and what it means for all of us.
Well, thank you everyone for another great year, and we hope to see you again out in the field.